Hello, welcome. This is biology. We're going to go over chapter 17 in the textbook Biology 2E by OpenStax, and that's going to cover biotechnology. So we'll talk about biotechnology, genomes, genome sequencing, genomics, proteomics, cancer, all that in this chapter. So let's go ahead and get started. We're going to start off, of course, with 17.1, and that's going to cover biotechnology. So we're going to look at some of the devices and procedures and how we analyze DNA. So we'll look at gel electrophoresis, and we'll look at molecular and reproductive cloning, how they're different, and then biotechnology in medicine and agriculture as well. So humans have been manipulating genetic material for almost their whole life. This wolf, we've taken the wolf and we've made dog breeds out of it by selectively breeding. We've done the same for crops such as this one. We've made all these different types of vegetables off of one initial crop. And then same thing with corn. Wild corn used to look like that, and we've just continued to breed it so that we get corn that we now can recognize that's in the grocery store. So this is selectively breeding, and we've been manipulating DNA for thousands and thousands of years without actually getting into the nitty-gritty part of it, which is what we're going to look at today. One major breakthrough in biotechnology was the discovery of penicillin. And so Alexander Fleming was the scientist, the researcher who was discovering ways to get rid of bacteria. So it was a problem. He worked in a hospital lab, and he went on vacation. When he got back from vacation, his petri dish of Staphylococcus bacteria had some mold growing on it, and that happens. You leave a petri dish in the open, mold is going to grow on it. But what he looked at was where the mold was growing, there was no bacteria growing. And so he named it penicillin. He named the mold penicillin, and he was able to create the drug that we know today, just based off of that little discovery. Other modern advances. So insulin is a huge modern advance of biotechnology. We don't have to make it in pigs. We can make it in the lab now. The flavor saver tomato, it has a longer shelf life. So this is a gene that's put into things that we eat, and it helps them last on the shelf a lot longer. They don't decay as fast. Some other things like how far is too far, nutrition value, are we harming ourselves? There's always bioethics to consider when trying to manipulate DNA. In this case, here's a little comic that says the ideal baby, and you can choose what you want your baby to look at based on different genes to pick out. So it can be good and it can be bad based on how you feel, based on the environment, and so on and so forth. All right, looking at this picture, what is wrong with it? Well, we can't manipulate DNA with our hands. It's too small to manipulate. This is obviously a very bad stock picture. But we have to so we can't manipulate it with our hands. We have to find other ways to manipulate it. So we're going to have to develop some procedures and make some new machines and all that, and how to manipulate DNA. One interesting way, or one way that we can extract DNA, so we're not going to manipulate it just yet, but we can extract it. So here's a bacteria cell. Bacteria has all this DNA floating around in the center. We can easily disrupt the cell membrane and then extract each piece of the cell if we centrifuge it. So a centrifuge is this contraption that spins really fast, and it separates particles, whatever you put in the tube, by weight. So heavy things are going to go to the bottom, and then lighter things are going to float to the top. And so we can extract DNA simply by doing that, by spinning it in a centrifuge, and there's DNA and other things floating up here, so it's not perfect, but that is one way to do it. And you can do this at home too. So I can show you the procedure, but you're going to have this like strawberry. You could do this with strawberries or bananas. They have a lot of DNA. You can lyse the cells, the strawberry or the banana cells, and you are going to not spin it in this procedure, but eventually you get this like spaghetti-like material that's DNA. And so this is the whole procedure on how to do it. You crush your strawberries, you add soap, water, and salt and stir. There are some like amounts for each one of those. There's a nice YouTube video here, or you just search it on YouTube too. And then you'll stir and you pour it through like a coffee filter into a tube like this. So if you have a test tube, great. If you have something long and skinny like that, that's just as good. And then you're going to slowly pour some alcohol over top and you create this like layer. And then in between the alcohol layer and then the strawberry goopy stuff at the bottom, you're going to disturb this layer with like a toothpick, or if you have a kebab stick, that would even work better. And you're going to disturb that layer, and then you'll see some like stringy stuff start to form, and you can kind of like pull it up on the stick, and that's DNA. So just easy enough to do at home. So some other parts of DNA technology: gel electrophoresis, PCR, molecular cloning, reproductive cloning, gene therapy. These are all things that we're going to look at here. First, we'll go over electrophoresis. So electrophoresis is pretty neat. It's a way to separate DNA based on its size. So we don't just like load DNA into here. We have to do some things first to it. But DNA is going to be broken into chunks pretty much before we put it into these wells. They're called wells. They're little spaces. Once we load the DNA into the wells, it's dyed so that we can see it, because it's usually invisible or you can barely see it. And then this gel is placed into a liquid buffer, and there's some electricity that gets added to it. And so since DNA has a slight negative charge, it gets repelled from the negative side when we put electricity on this, and it pushes the little fragments towards the positive side. And so as these little fragments move through the gel acts kind of like a filter. So smaller fragments are going to be able to get through the filter faster, so they will be closer to the other end. And then big fragments, big long DNA fragments, are going to get hung up on the filter, so they're not going to move as quickly. And so that's how these DNA sequences are arranged. So, really long sequences are back here, closer to the wells. Shorter sequences are up there. So gel electrophoresis can be used in determining like parental lineages. If the child is in fact the mom and the dads, this is one way to do it. Forensic science as well. Here's a real picture. So here are the wells. On the sides here is what we call a DNA ladder, and those give us relative base lengths or sequence lengths, so that we can estimate what the sizes of these guys here are in the center. But this is the actual DNA that was pushed through the gel, right? And then you look at it on this little box here, and that can show you the DNA UV light. Okay. So here's a little gift from Amoeba Sisters that's really cute, I think. So we load the DNA into these wells, and here's the baby guppy, mother guppy, father guppy. So we're just kind of looking to see, oh, is it the father or is that the mother? Well, yeah, because this baby guppy has base pairs that are found in mom and dad, or sequences, lengths that are found in mom and dad. So yes, the baby is in fact both mom and dad's there. Okay, so answer this question. In gel electrophoresis, it separates molecules by movement due to size and electrical charge. Using the diagram above, what can you say about the molecules that will move the quickest through the gel? So molecules that move fast are going to be what? Smaller. Molecules that don't move as fast are going to be larger. So that should leave us with at least two answers. So the quickest ones will be small and small. So now what are we going to say, negative or positive? So DNA is negative, and so that's going to be A. A should be your right answer. All right, PCR is another really neat method to get more DNA based on what you were given. So a lot of the times we need to make copies of the DNA sequence that we have because we only have a little bit of it and we want to do a lot of work with it. And so if we only have a little bit, we can't do much work. That's where PCR comes in. So we load these little wells in here. It's a little tray with a bunch of wells. We add some different chemicals to it, and that's going to be like an amplifier and so forth. But at the end of PCR, we're going to double the amount of DNA that we started, and we can let it run for many, many, many cycles, and each cycle will double the DNA. So you'll end up getting a really large amount of DNA at the end. But really great for studying like disease causing fragments, paternity, and then like forensics as well. So this is how it happens. That's our original sequence. We're gonna just replicate it, and then we end up with 2 strands, and then 2 strands turns into 4 strands, and 4 strands turns into 8 strands, and it just keeps going from there. So you're gonna end up with a lot. All right, cloning. There's 2 types of cloning. Molecular cloning, which is just cloning a segment, and then reproductive cloning is cloning a whole organism. So this is a little Dolly the sheep here. So let's take a look. Molecular cloning is used to reproduce desired regions of fragments of DNA. So they manipulate and study specific genes. So here's a plasmid. If you remember back from when we went over cells, plasmids are small circular segments of DNA. So it's not a huge genome. It's just a small bit inside usually a bacteria. And on plasmids, there's usually genes for like antimicrobial resistance, or like antibiotic resistance, right? So they're going to have these target genes. So it's like, in this case, the insulin gene. And we're going to want to reproduce that and put it into a different plasmid or into a different organism or into an organism's genome. And so that organism can produce insulin. Plasmids, so we're going to take them from the bacteria host, and then when we put new DNA into another host or into another molecule, it's called recombinant. And so the proteins that we are expressing from that DNA are called recombinant proteins, right? So we have DNA, DNA has genes, genes code for what? Proteins, right? So at the end, we get these new recombinant proteins. So an organism that could maybe not make insulin before, once it receives this recombinant plasmid, with the new gene that we put in it, it would now become an organism that could create insulin, in this case, for this example. Okay, so reproductive cloning, a little bit different. We're not talking about just cloning like a segment of DNA or just one gene in that case. But in this case, we're going to recreate a whole organism. So many, many, many genes have to be copied. We could do this pretty easily by taking an egg from a donor, and we will take it and make it a diploid nucleus. So remember that egg cells, they're haploid, right? They're sex cells, and sex cells have half the amount of DNA in them, right? So when they become a diploid nucleus, when they meet up with another set of DNA, they become diploid, and then that tells the organism to start growing into a zygote. So now when they take DNA from a somatic cell, so they take a diploid nucleus from a somatic cell, and they replace a diploid nucleus with the haploid nucleus in the zygote. So they swap them out. So the DNA in that somatic cell now is just the DNA of the original organism. So the resulting organism becomes the genetic clone. So it would be like taking like a skin cell and taking the nucleus from a skin cell in your body, putting it into an egg, and then since that DNA is the exact same as your DNA from your body, you get a whole clone. So Dolly the sheep was created in this way by reproductive cloning. A egg donor was the Scottish blackface sheep, so you can see it's a different type of sheep than Dolly was. But the egg cell was taken from this sheep. They removed the nucleus, so it's just a blank egg with no nucleus in it. The mammary cells, these are the cells that they used to get the diploid nucleus out of. They took the diploid nucleus, they put it into this now empty egg, and because now it was a diploid egg, it started to develop into a zygote. And so they inserted the zygote back into the surrogate, and the surrogate gave birth to Dolly when Dolly became full term. And so this was an exact clone of the sheep that donated the nucleus, not the surrogate mother. Genetic engineering is the alteration of an organism's genotype using recombinant DNA technology. And so the addition of foreign DNA in the form of recombinant DNA plasmids is basically what it is. The organism receives the DNA, and so it's technically known as a genetically modified organism. So those flavor saver tomatoes receive that flavor saver gene that lasts or causes them to have a longer shelf life, and so they are technically GMOs. Are they usually harmful? No, but we still need to do more research just to make sure that that's correct. Transgenic organisms have DNA fragments from different species, like so Roundup Ready soybeans, borer resistant corn. So these two crops, soybeans and corn, they received some genes from other organisms that were already maybe like immune to Roundup or immune to those borer bugs. And so they were inserted into their genome, and now they naturally are resistant to these things. So it is better to be naturally resistant, say, to like an insect, because then we don't have to spray pesticides. And we could really go down a rabbit hole here with talking about these things, but that's the basis of it. And here's some cute like comics I found on the internet. So this one, we're growing ears on mice to be transplanted onto humans. When you play with Hox genes, if you know anything about them, are genes that can be repeated over and over again to create more segments. So the spine is one area where we have Hox genes. So normal spine in like maybe like a dog, I want to say. And then obviously snake, right? So Hox genes, right? And you get limbs to pop up and tails to pop up wherever. And here's another example of that with a leg here instead of an antenna. Okay, so gene therapy, it is pretty interesting. This is a way that we can introduce a good gene into an organism by way of a virus. So we put the correct DNA that encodes for a gene. So let's say like you have cystic fibrosis, which is a horrible disease of your respiratory system, and you don't create mucus properly, or you create mucus, but it doesn't have a way to get out. Like it's too sticky and it clogs up your airways. That's basically cystic fibrosis. It's a very vague overview, but anyway, we use gene therapy to introduce a virus or introduce a gene through a virus. So it infects your cells, and what happens when a virus infects your cells. It injects its DNA into the cell, and that cell starts producing the proteins that that gene encodes for. So we can use an adenovirus, which is what this guy is here, and we can cure people with defective genes that way. Transgenic organisms, so again, they have DNA from other species. DNA is DNA no matter what the organism is, so it doesn't matter if they received it, if it's a corn species and they received it from a soybean species, it's still, you know, ATCG in different forms or in different different patterns, but it's still DNA, so it's going to code, that new organism is going to code those genes and those proteins. Targeted genes are from organisms that naturally express a certain trait. So like I said earlier, pesticide resistance, if an organism is naturally resistant to like a borer insect, then that's great. We're going to target that gene, remove it from some sample cells, and then try to inject it into a new organism, which would then be a GMO or a recombinant organism. So pesticide resistance, nutritional value, like golden rice, that's one to look up. Shelf life, like a flavor saver tomato. Color pattern we can do, texture, how many are produced. So like corn creates 2 ears per stalk. Would be great if we could double that or triple that and create more per stalk. So these are the types of things that we're interested in just for agriculture purposes. So GMOs, answer that. GMOs are created by what? Generating genomic DNA fragments with restriction endonucleases, introducing recombinant DNA into an organism, overexpressing proteins in E. coli, or all the above. So your answer should be B in that case. All right, so that's the end of 17.1. That's the longest section in chapter 17. So you should now be able to describe how gel electrophoresis works, and you should be able to explain the difference between molecular and reproductive cloning and how we use biotechnology in medicine and agriculture. So next we're going to take a look at mapping genomes. And genome or genomics is the study of an entire genome. So a genome is the complete set of genes, complete DNA, and all the nucleotide sequences and how they're organized within that genome. A genetic map is an actual map that maps the locations of genes on chromosomes. So humans have their own, we have created a genetic map for humans, for Homo sapiens, and we can say which genes are on which chromosome and where exactly they are on that chromosome. Genetic maps, again, so they aim to determine the location of genes, estimate the distance between genes and genetic markers based on recombination frequencies. So we talked a little bit about this during the genetics section and what a recombination frequency is. So you always see red hair with freckles, right? So those genes are probably located pretty close together, so they don't get recombined very easily, so their frequency would be low. But you don't typically see like blue eyes and black hair, right? So that's probably going to be really far apart. Physical maps, they of a genome, they provide detailed information about the physical distance between genes. So a cytogenic map, a radiation hybrid map, and a sequence map are all different types of maps that we could find, or that we have for these things. So let's take a look at a couple of them. So a cytogenic map is a map that shows the appearance of a chromosome after it's been stained and viewed under a microscope. So this picture on the left here is just a computer generated one. And then these down here are actual pictures of those chromosomes. So they're all condensed so that we can see them and easily pick them out. So it's possible to determine approximate distance between genetic markers using a cytogenic map, but maybe not the exact distance, right? So we could say like, oh, you know, they're pretty close together, but we can't say exact number of base pairs, so we can't count that. So answer this question. So information obtained by microscopic analysis of stained chromosomes can be used in, should say D there for your answer. Okay, so that was the basics of mapping genomes. So we can map genomes and figure out how many base pairs or how many lengths away we are with the with genes. I know we only talked about cytogenic map, but radiation hybrid map and sequence map go more in depth, and they will tell you more about how many base pairs apart each gene is. But the cytogenic map is nice to look at, right? Okay. Whole genome sequencing. So there's 3 types of sequencing, and we're going to look at whole genome sequencing in particular here. So whole genome is a process that determines an entire genome's DNA sequence. So what bases are in what order. So we're going to go through this strand of DNA over here and say, well, that's A, and then G comes next, and then another G, and then like an A, a T, a G, a C. So we're going to go through the whole genome and determine what letter comes where, what nucleotide base comes where, and in what order, and how many there are. That's a whole genome sequencing. It takes a really long time to do it, but several laboratories now actually provide the services to do this. We can use it to treat genetic disorders. So using your whole genome sequence, you can determine what genetic disorders you are predisposed to, or if you have any currently. Biofuel development has been used in agriculture, pharmaceuticals, pollution control. Basically, you name it, we're going to try to use it to solve some problems. In 2010, okay, so whole exome sequencing is a little bit different from whole genome sequencing. So whole exomes are just going to be the usable parts of DNA. So it doesn't take as long. It's a little bit easier to use and understand. But whole exome sequencing was used to save a 15-month-old boy who was suffering from Crohn's-like symptoms. So Crohn's disease is an intestinal disease, but it wasn't actually Crohn's. So using this method, doctors were able to diagnose a defect in the boy's DNA, which was causing apoptosis in his intestinal cells. So apoptosis is cell death. So his intestine was basically just dying. As soon as new cells were created, they would just die. So a bone marrow transplant was conducted, and so the boy received the correct form of the gene, and he was actually cured. So he grew up and he's living a normal life now. But this is a reference here. So this is the reference of the normal, this is the child, and this is the mother. And then these letters that you see here are the nucleotide bases. So the defect is right here. So he has an A instead of a G, which would be a normal coding. But the mother, if you notice this green line here, she carried both forms of the gene. And so that's how the child ended up getting that disease. So just one base pair mistake, one mutation causes that whole problem. Okay, so modern methods of sequencing utilize the chain termination method. It also has another name, but it sequences the DNA, and we can figure out what order they are based on this method, because you can't just look at a strand of DNA and say, oh, that's A, and oh, that's G. You have to do it by, manipulating it in different ways. We can't touch it to manipulate it, so we do it this way. One main method is shotgun sequencing, and the other two, pairwise end sequencing and next generation sequencing, are just like shotgun sequencing, but they're a little bit more effective and efficient. But basically, what happens is that these cloned genomes, so we probably put them through a PCR machine and got some more copies. We're going to take those and shoot, like a shotgun, different splicing base pairs of DNA. So it tells you when to stop coding. And so that's going to split the DNA into sections, like you shot it with a shotgun. And then based on these sections, you're going to piece them back together like a puzzle. And then this piecing back together part of the process is where they start to determine what base pairs are where. It sounds super complex, and it is, but that's the basis of it. The things that are loaded into the metaphorical shotgun are called dideoxynucleotides, and so they are super similar to our deoxynucleotide, except that they have this missing O there. And so when a dideoxynucleotide is incorporated into DNA, the synthesis stops. And so that's where we get all of those pieces that are cut. So each one of those has a dideoxynucleotide on the end. And so that's what creates all those segments. These segments are then run through different protocols, but we can determine how many of each base are where, or not how many, but which base is located where, using those dideoxynucleotides. So the DNA is then again separated based on its size and the bands, and then we can determine what sequence is there. So what is the overall goal of whole genome sequence? So we want to determine whole genomes of different organisms so that we can do research, but not necessarily on the organism itself, right? We want to be a little ethical about it. So it's better if we just have the genome sequence so we don't have to use living organisms. The first genome that was sequenced was Escherichia coli, so E. coli, the bacteria. The second one was Saccharomyces. It's a yeast fungi. And then since then, we've also done some other ones. So mouse, fruit fly, nematode, and humans. These are what we call model organisms, and they are the species that we use to understand biological processes in species that are similar to them. So if we take the next course of biology, general biology 2, you will learn how these organisms are very different from each other. And so we can use them to determine how different genes are going to react, how different maybe diseases react in different organisms and so forth that are closely related to each one of these. Genome sequence uses DNA microarrays. So microarrays are used to detect gene expression and are commonly used in medicine. And so DNA is affixed to a glass slide or a silicon chip, and the process just identifies active genes. So this one, genome sequencing, or DNA microarrays, DNA microarrays are a lot easier to create because we're only looking at active genes instead of the entire genome. So we have a lot of our genome that doesn't actually code for genes. It's just kind of nonsense. And so this process, we're just looking at the stuff that makes sense there. So that's just another way. So whole genome sequencing can be used for advances in all of the above. So medical, agriculture, biofuels, all of those. So that's 17.3. So you should be able to describe the 3 types of sequencing, or at least name them. I'm not going to ask you to go into detail there, but you should be able to tell me what whole genome sequencing is and why it's important and what we use it for. All right, 17.4, applying genomics. We're going to look at pharmacogenetics here. So this one's pretty short. Pharmacogenetics and applying genomics are going to be very useful in personalized medicine and predicting diseases, and also drug and if you had a transplant, interactions, or reducing an interaction. So based on your genome, you are able to determine what's going to work for you, right? So it's not going to be like a one size fits all type of thing. It's going to be one size fits you if it's how your body works. And so pharmacogenetics evaluates the drug effectiveness based on your genome sequence. It's a new thing. It hasn't really been incorporated yet because, again, we have to code your entire genome, but it is something that maybe we could do in the future. Again, one dose fits all, so we're not going to just be like, name brand ibuprofen. It's going to help you. Well, you're going to take your own certain type of ibuprofen to help what you need. So it eliminates a lot of trial and error. So a lot of the times when you get diagnosed with like a new medicine, say for like blood pressure, they're going to start you on a basic one that they start everybody on and see how you react to it. Well, if you go back in a few months and your blood pressure still hasn't dropped, well, they're going to try you on a different one. So it's going to reduce a lot of that trial and error and that waiting time in between to see if you're reacting to the drug like you should be or you're not. Okay, so it also can be used to determine or detect potential toxic effects of a drug on a particular person, especially like allergic reactions. Easy for me to say. Allergic reactions to drugs for a particular person. So that would be nice to know before you take the drug and then have, a whole reaction to it. Metagenomics involves isolating DNA from multiple species within an environmental niche instead of a pure environment. So metagenomics is mostly used to speciate bacteria based on their genome. It's used in the lab to determine what bacteria are reacting to certain effects of whatever the researcher's doing. Basically, you can't grow bacteria, just one species of bacteria at a time. It's really difficult to. I mean, you can do it, but it's difficult, and they grow better if you have more types of bacteria. So they create this like film and this like microenvironment. So metagenomics makes it easy for the lab technician to speciate the bacteria, and it just takes their genome out, and using their genome you can determine what species that bacteria is. So answer that question. Genomics can be used on a personal level to do what? Well, D, right? Decrease transplant rejection, predict genetic diseases, determine the risks of genetic diseases in children. So D, all the above there. Okay. And lastly, 17.5, Genomics and Proteomics. We're going to look at systems biology. We'll look at what a proteome is and protein signature. So let's start with proteomics. A proteome is a full set of proteins produced by a cell. So your cells in your body, they're not going to produce all the same types of proteins, right? The cells in your brain, the cells in your gut, the cells in your skin, the cells in your muscles, they're not going to produce all the same types of proteins. They're going to produce the proteins that that cell particularly needs. And so proteomics is a study of a proteome's function. So it's a study of what the proteins that are being produced by that cell. It's the study of what they do for that living organism. So it's super dynamic because it's just looking at particular cells and not a whole organism. It relies a lot on protein analysis, and is used to study mostly different types of cancer. So cancer produces certain types of proteins, and it usually overproduces those proteins. And so being able to determine and find these different protein signatures in a person's body would be super helpful in detecting cancer, especially cancers that are really difficult to detect. And then metabolomics, this was just a vocab term that was in the textbook, but it's just the study of molecule metabolites found in an organism. So that's also useful in determining and finding cancer in a person's body. So metabolites are those things that are produced when you metabolize like molecules, so like such as glucose. So what's produced after you create ATP from glucose metabolism. So it's like that. Protein analysis, there's some different ways that we can analyze. Mass spectrometry is the major overall one. It identifies molecules' characteristics. X-ray crystallography determines the crystal structure of a protein, so what it looks like at the atomic level. Nuclear magnetic resonance determines protein structure, and protein microarrays enables the study of protein interaction. So that one's a little bit more useful on the how do they work type of level. Okay, so two hybrid screening is just one way that scientists use to figure out if proteins interact. So if a protein interacts like you see here, then a gene is being transcribed. But if the proteins don't interact, then a gene is not transcribed. So that's the like yes and no if the proteins do interact or not. This is really helpful in determining systems biology. So, proteins are the basis of what DNA codes for. And so proteins are basically what makes up an organism. Most pharmaceutical companies and research companies, they target the proteins that are produced in the person's body. So this is going to be super helpful to them in how they develop new drugs and understand different mechanisms of action, and so that they can develop new classes of drugs even to help people. Cancer proteomics researchers are going to look at a patient's genome to understand the basis of disease and support early detection. So biomarkers, what are those? They are the individual's proteins with altered expressions. So they often produce false negatives. So a protein signature is created and the entire sets of proteins have altered expressions, and those ones are more reliable. But both of these are going to be used in determining and catching whether the patient has cancer or not. And it really helps with early detection and improved screening overall of cancer. So what is a biomarker? A biomarker is a protein that is uniquely produced in a diseased state. So it's going to be a weird protein. And that's it. So 17.5, you should be able to explain systems biology, which is the organism that produces proteins and how proteins interact with each other. Proteome is all the proteins that the organism produces, or the cell. In this case, the cell produces. And then a protein signature is a set of proteins with altered expressions. So that's chapter 17, biotechnology, and I hope that was helpful.